All right, Chapter 6 and 7 today. Chapter 6 is on specimen collection and processing, and then we'll talk about, what's Chapter 7? Microscopic morphologies. And then in lab today, we're gonna begin doing gram stains and colony morphology. So we'll get to learn how to characterize organisms, which is one of the first steps that we do when we're looking at a culture and we're trying to determine what the pathogen is. You characterize the colony morphology and you do a gram stain, and that's going to tell you which pathway to follow with your biochemical testing. All right, so specimen collection and processing. This is a lot of, it might seem kind of detail-oriented and useless material, but it is important for us because it determines how well we're able to do our job. And unfortunately, we're not the ones collecting our samples. So even though we know how to properly collect a sample, we're not the ones doing the collecting. It's usually the nurse or maybe even the patient doing it. But we still have to know how to properly collect a sample. If it's poorly collected, then that means basically what we're working with is garbage. It's contaminated with normal flora on our skin and other things that can sort of hide the pathogen. It makes it difficult for us to recognize what is the pathogen and what is normal flora. So definitely go through these objectives when you're studying for the exam. All right. This case in point is talking about a microbiology lab in a physician's office. They noted that a cervical sample was submitted on a Gembeck auger plate. And Gembeck auger plates are specifically designed to isolate gonorrhea. Gonorrhea is a fastidious organism which is hard to grow. It requires some supplemental nutrients in its media, and that's what Gembeck auger is. So the physician's office collects the sample and plates it on this Gembeck auger and then transports it to the microbiology lab. So the technician begins streaking through the area of inoculation on this Gembeck auger plate and notices that the media is dry in appearance. And then further evaluation, they noted that the medium was expired. So because the media is expired, it's dried out, the nutrients are probably not at their best, you're compromising the results that you're getting if you're not using media that's in date. That makes sense, right? I think Ms. Grimmie's probably going to drill into you using things that are in date, checking your expiration dates. It's all part of regulations that we follow, that what we're using is in date. And if it's not used a lot of times in physicians' offices, they're given these media and they don't use them all the time and they don't check the expiration dates because they're not as well trained as we are. And so you'll get media all the time from them that is expired, in which case, what do you think you should do? Go ahead and process the culture. You've got to notify somebody, right? Definitely call the physician's office and tell them, that there's a problem with the sample and it needs to be recollected. Now, the physician may be adamant about processing the sample anyways, and in that case, you would definitely put a note in there saying that the results are compromised because of a certain, because the reagent or the medium was expired. Okay, so there's basic principles for proper specimen collection for microbiology. You've talked about proper specimen collection in hematology, I'm sure, labeling the sample, when to collect it, where to collect it, how full the tube needs to be. Does it need to be clotted or not clotted, right? So microbiology is a little bit more in depth. Because we are dealing with infectious processes, we need to collect the sample before antibiotics are given to the patient. Those antibiotics can potentially affect if we are able to isolate a pathogen or not. They could even, in the case of blood cultures, if the patient's given intravenous antibiotics, the antibiotics could be in the blood that we collect, and that could inhibit the organisms from growing in the blood cultures. And so we'd get a false negative result because antibiotics were started too soon. So specimen collection should be taken in the acute phase while the symptoms are active of the disease and before any antibiotics. Select the correct anatomic site. If a patient presents with symptoms suggesting a urinary tract infection, you're not going to collect a swab of the throat, right? So that's important. Believe me, it happens. Proper technique. So collecting a sample with as little contamination of the normal flora as possible. When you're collecting a blood culture, you sterilize the skin very well before you collect the blood culture. And that way you don't contaminate the blood culture with skin flora that's normally present on your skin. Or, for example, when you're collecting a urine sample, there are steps to cleanse the genital area, and you collect, ideally you collect the midstream portion of the urine in the early morning in order to avoid contamination from normal genital flora. Collect the appropriate quantity. If you're not given the correct amount of sample, the probability that you can isolate a pathogen is severely reduced. Labeling the specimen accurately. Who collected it, when they collected it, what the source is, all of that has to be on the sample label. Pack to maintain viability and prevent leakage. You want to securely fasten the lid. Pack it so that if it does leak, it's not leaking all over the person transporting it or all over the paperwork. Maintaining viability means that the organisms are still viable. They're still able to grow by the time it gets to the microbiology department. Physicians' offices may collect samples early in the morning, but the courier may not pick them up and deliver them to the micro lab until late in the day. So it's important that the proper transport media is used for transport containers, and that all depends on the type of sample that you're collecting. Transport and store specimen promptly. The sample may require refrigeration to preserve it. It may, you certainly want to avoid any high heat temperatures. Notify lab in advance if unusual pathogens or bioterrorism agents are suspected. So usually we set up routine types of media depending on the site of collection, and we'll talk about that in a minute. But if the physician is suspecting a very unusual pathogen, it's important that the microbiologist is told that fact, because we may need to set up additional media. For example, Legionella causes respiratory diseases, and it's not isolated commonly on our respiratory media that we set up. So if the physician suspects Legionella, it's important that we know so we can set up a BCYE plate in addition to the normal routine auger, and that plate isolates Legionella. A bioterrorism agent, if you think your patient has Ebola, it's very important that we know this, right? So we can protect ourselves and the co-workers, and so that we can handle the sample properly. Okay, so collection procedures. Of course, you want to use a sterile container for any kind of sample that you're using. We can't have it contaminated with environmental flora, because that's going to throw off our results, right? The one exception to this is stools. Stools can be kept in a clean, leak-proof container. People have brought them in Tupperware, paint cans, clean paint cans. Swabs are probably the most commonly collected microbiology sample, but they're not the best. It's very limited quantity, and it's prone to drying out. Typically, they collect swabs on like wounds or throats, nasal, pharyngeal, those kind of things. And a lot of times it's it's not bad. It's just it's the easiest thing to use. So upper respiratory tract, ear, eye, genital tract, your swabs, it's pretty unavoidable to use a swab. But other things like wound cultures, bed sores, surgical site infections, the ideal specimen of choice would be an aspirate of the wound. But swabs are the most commonly collected. So what the swab is made of is important. Cotton, it can be toxic to some types of bacteria. So we avoid cotton. We usually use this Dacron or calcium alginate swabs. And then we saw yesterday a variety of, or not yesterday, last week, a variety of different types of swabs, and they can have some preservatives in the bottom to help keep things moist and viable. Okay, so lesions, wounds, and abscesses. If we're going to collect these, the wound site, if the sample is labeled with simply just wound, that's not acceptable, although it's very common. We don't know what that site is. A wound can be anywhere on the body. So we need the exact anatomic site. A wound collected from a genital site is going to be very different from a wound collected from the foot. It has different normal flora depending on the anatomic site that is collected. So it's important that we know where exactly that swab came from, or that specimen. Ideally, it's an aspiration. It could be a swab, but you want to collect from the line of infection. So that's like the outer edge of the wound. You don't want to collect from the middle of the wound. And a lot of people want to go there because it's nice and juicy and nasty looking, but that's the oldest part of the wound. And it's a bunch of dead debris, dead organisms, dead white cells. So you want to get what is alive and spreading. So that's the outer edge. And you want to clean the area to remove any normal flora, any contaminants that are present. So you want to clean the wound first and then collect the sample. And then place it in a sterile tube or vial. Now, patient-collected samples, these are pretty common, actually, where the patient is instructed to collect the sample. And unfortunately, we're not involved in this either many times. But the nurse is supposed to explain to the patient how to collect the sample. And they may use technical terms. They may completely avoid describing the process altogether. So it's important that we give the patients instructions, not only written, but also verbally and also with pictures to help them understand exactly how to collect the sample. So one of the first types and the most common types of patient-collected samples is your urine, your clean catch midstream urine specimen. So ideally you want to get it in the morning. You want to cleanse the external area to reduce the normal flora that's present. The first part of the urine flow goes into the toilet, and the middle portion of the urine is collected in the cup. And that allows for the urethra to be rinsed. And this procedure is similar if you have to catheterize a patient to collect a urine sample if they can't go. The nurse should be doing the cleansing and collecting the middle portion from the catheter. Sputum is also very commonly collected patient sample. Kind of difficult to collect. You want to get a first morning specimen. They've been laying in bed for several hours. Things have built up. It's very commonly contaminated with oral pharyngeal flora. What we want to avoid is the saliva and the nasal secretion, just the spit that's in the mouth. What we want to get is a deep loogie, deep in the chest. So an expectorated sputum has a patient rinse the mouth with water, spit it out, and then a deep cough to produce the loogie. An induced sputum can also be collected, and that's where they inhale a substance that stimulates them to cough. Oops. And then finally, stool. Stool is collected for the identification of gastrointestinal pathogens. They might collect samples over 3 days. Sometimes you might get 3 samples in 1 day, and it depends on your facility on how they handle that. But ideally, you want one over 3 days, 1 day each. For parasites, also, you'll collect 3 samples, and this may take over 10 days collection period. Antigen screens, you might use a little cassette to detect for gastrointestinal pathogens. In that case, only 1 sample would be required. To remember with stools, you don't want to contaminate the stool with urine. The patient might dip it out of the toilet. You want to refuse those samples. They're contaminated. Also, if it's contaminated with barium contamination, which is something that they drink if they had to take a CAT scan, their stool has this chalky white stuff in it. So you want to wait until that barium goes through their system after a few days and then collect the stool sample. And commonly, the stool samples are placed into different preservatives. There may be 3 to 5 different vials in a stool kit, and you'll separate out the stool into each of those vials. And they each do something different. They might look for, one might be for a culture, bacterial culture. One might be for ova and parasite exam, a microscopic exam, so it preserves the morphology of eggs and worms that might be in the stool. Another one might be for immunoassay testing, or Clostridium difficile testing. So it just kind of depends on what the doctor's looking for, and we'll take a look at some of those containers in the lab. So this is just an example of some collection guidelines. I definitely want you to know how to collect a urine culture properly, a clean catchment stream, the stool and sputum. Those are the most important. The rest are kind of common sense. So if you need to collect a spinal fluid sample from a person, basically all you need to do is disinfect the skin and use a sterile needle and collect an aspirate. So anything that's going, any kind of sterile site or sterile fluid that you're collecting from a patient. Blood cultures. Who here has collected a blood culture? I know we had a couple of phlebotomists. So blood cultures are glass bottles that you would collect. So when you're doing a venipuncture, instead of collecting a tube of blood, you collect this bottle of blood. And the bottle has media in it, which will grow any kind of organism that's present. And then you stick it in an analyzer, and it can detect whether or not bacteria is present. And it's a little more technical than that. And we'll talk about those again soon. So it's just examples of how to collect certain types of samples. Okay, so once you've collected the sample, the next thing that you should do, or should be done, because you're probably not collecting it, you want to label it with your patient identifiers. You want to put the culture site on there, the source of the sample, the date and time of collection, and the collector's initials. In case there's any problem, we can contact who collected that sample. A test requisition. Anybody hear that before? Test requisition. Samples aren't collected unless we have a doctor's order. So you want to match the test request with the sample that you're provided with. So the test request should have it has a variety of different information on it. It should have the patient's name, date of birth, gender, maybe where they're located, who ordered it, the site or the type of test that they want, a urine culture, stool culture, wound culture, the date and hour of specimen collection. That might be important. The physician may want a sample collected at 7 a.m., not just any time through the day. So it's important that we take that request, that test order, and match it to our sample and make sure that they coincide with each other. Some safety to consider when you're transporting these samples. You want to make sure that they're in a leak-proof container and then in a secondary container. So the primary sample is the cup or the swab that's holding the sample. It needs to go into some kind of sealable secondary container like a bag. Papers should be kept separate in case leaking does occur. Specimens shouldn't contain needles or sharps, so if it's an aspirate, they should have removed the needle and put a cap on the syringe and not given you a needle and syringe. And then all specimens should be handled with PPE and necessary engineering controls. So if you're transporting a spinal fluid, you should be wearing gloves. If you're transporting stool, they should be wearing gloves. Transport within 30 minutes or within 2 hours, and this may change depending on where it's coming from, and the laboratory can take into account those kinds of situations. For example, if it's from a physician's office, they'll instruct the nurses to place it into the refrigerator until it's collected, or place it on ice or in a freezer, or collect it in these particular containers that contain a preservative, whatever the sample or whatever the case may be. Now the specimens, they need to be inoculated onto the media immediately, if possible. Sometimes we get busy and we can't process microbiology specimens as quickly as we should. So in that case, we need to store them appropriately until we can process them. So certain pathogens can be stored at certain temperatures. You don't want to put a spinal fluid in the refrigerator because spinal fluid pathogens are harmed by the cold. So it just depends on your facility, and they'll have it outlined of how to store a specimen before you process it. Okay, so here's some guidelines. These are different types of specimens that should be refrigerated if there's a delay in processing. Your sputum, your urine if it's unpreserved, your feces if it's unpreserved. If they are preserved, then they can sit at room temperature. The preservative is doing its job. Body fluids, spinal fluids, abscess, lesions, wounds, those things should stay at room temperature. Okay, there's a variety of different preservatives that can be used. An example for urine is boric acids. The goal of preservatives is to maintain viability and at the same time keep organisms from reproducing. So we want to maintain the count, the colony count, or the count of organisms present in that sample that was present in the patient. Okay. There may also be some anticoagulants in certain transport systems. Heparin can be used. SPS is common for like blood cultures, those kind of things. So. Transport media. Again, this is the main thing I want you to know. Transport media does not promote growth, but it preserves them. It ensures viability. Okay, so Ames, Stewart's transport media, those are some examples. Here's a Gembeck medium. So it grows Neisseria gonorrhea, it preserves it. And you put a little capsule in here which releases CO, it turns oxygen into CO2 because gonorrhea likes CO2. It's a capnophile, right? Capnophile. And then this is an example of blood cultures that you would collect. If they suspect bacteria is in the blood, we've got an aerobic culture, we've got an anaerobic culture, and a pediatric vial where you can collect smaller amounts of blood. Here's an example for packaging infectious substances. And if you're going to be required to package substances and mail them through the mail, FedEx and all that, you'll have to take a course to do this and be certified in it. But this is just an example of, you put the sample in there, there's an absorbance, and you put it in a secondary. There's a lot more entailed when you're transporting infectious substances through the mail. Okay, levels of specimen priority. When you're a microbiologist, you'll have specimens coming from all different places. There are different types, and so it's important for us to prioritize our work. We need to get the most critical, invasive samples processed immediately, and then let those other types of samples that are more routine and preserved and not critical for the patient, let those sit and get to those when you have the chance to do that. So an example here, the first thing that you would get if you have a lot of, and I have a test question on this, I'll give you a few samples to choose from, and which one is the sample that you process first. So you want to process samples that where the patient is at immediate harm. They need a result right now. Okay, so amniotic fluid, commonly collected if they think that an infant, not an infant, a fetus is at risk. They'll collect amniotic fluid, and if there's some kind of an infection there, they need to find out now. Otherwise, that fetus could die. Blood culture is if someone is septic, they need to know as soon as possible. So you want to process that immediately. Brain, I think that's pretty self-explanatory, right? Spinal fluid, what are they thinking if they collect spinal fluid? Maybe a meningitis, which is very critical for the patient, right? Heart valves, again, kind of self-explanatory, right? Pericardial fluid, what's that? Fluid around your heart, again, pretty self-explanatory. We need to get to those first, process those first. The next, unpreserved samples, of course, your body fluids, like synovial fluid or peritoneal fluid, those things would kind of come next. Drainage from wounds. So these are time sensitive because they're not preserved. They're not critical for the patient in order to live, or die. Quantitation required. So this is where quantity of the growth present is important for diagnosis. So a catheter tip, they're trying to find out if a patient's catheter tip, indwelling catheter, is contaminated or infected with something. Tissue, urine would go here. And then finally, anything that's preserved can wait, last. So unacceptable specimens and specimen rejection. How do we handle it? What do we do? Okay, so suboptimal specimens would be collecting a sample that doesn't match your request. So if you get a feces sample and you have a urine culture order, obviously the samples don't coincide. So you need to call someone and find out what the deal is. Inappropriate transport container or leakages. If the urine has leaked all into the bag because they didn't secure the lid, you would reject it. Quantity not sufficient. For TB cultures, you need like 5 mils of sputum, and so commonly samples will be rejected because there's not enough sample. Or feces is another example. There's not enough. Transport time longer than 2 hours with no preservatives. If it's 9 p.m. at night and you get a urine culture and the urine was collected 6 a.m. that morning, it's not good anymore. It's been sitting at the patient's bedside stewing all day long. Specimen received in fixative or wrong preservative. Sometimes the nurses will put it in a preservative in order to, preserve the sample. Or fix it in the wrong thing. I had a case where they collected a bone marrow and you're supposed to put it in Boeing solution, but the nurse put it in formalin. Well, you can't do anything on a bone marrow sample after it's in formalin. She ruined it, so we had to recollect the sample, unfortunately. So it's important that it's fixed or preserved in the right way. Something else, anaerobic cultures that are anaerobic culture for specimens in which anaerobes are indigenous or not common. So say an anaerobic culture on sputum, not common. It's a site where there's commonly, no, I'm thinking of that different. Indigenous means that's where they live, isn't it? Basically, if your order doesn't match where your site would come from. So if they ordered an anaerobic culture, but it's somewhere where anaerobes can't live, or if they order an anaerobic culture, but it's something where anaerobes are commonly found, like the stool, it doesn't match well. It's the wrong order. Specimen is dried up. More than one specimen in one day. They just keep sending you samples over and over again. Or they keep ordering blood cultures, which is common. One swab submitted and a whole variety of different cultures ordered. That's a QNS sample. An expectorated sputum that doesn't meet our criteria. I've talked in lab about how sputums have a criteria for acceptance and rejection. So if a sputum has less than 25 white blood cells per high power field and greater than 25 epithelial cells, it indicates that the sample is not a good sample. So epithelials indicate spit. Less than 25 white cells indicates not a good sample. There's not a lot of infectious agents there. Another thing is if you get the culture and you see 3 different colony morphologies or more on the culture, that indicates a mixed culture. So we would reject the sample. How do we do that? You want to call for recollection. Call the nurse. Tell them about the sample not meeting the criteria or it's contaminated, and request a new sample. You never want to discard this sample until the issue's resolved. Document the reason for rejection either in the computer or some kind of log that's present in the lab. And then if the physician adamantly requests that the culture's performed anyways, you want to make sure you put a note in that culture report that the results are compromised. That sort of gets the responsibility off your back onto his back or her back. Okay, so once we've collected the sample, we've noticed that it's good. It's a good sample. We want to kind of do a macroscopic observation. So that's just looking at the sample as a whole. Is it a swab or is it an aspirate? Depending on whether it's a swab or aspirate, that's going to tell us how we're going to set up the plates. All right. Consistency of the stool. Is it formed or is it liquid? We're going to look for certain things if it's formed and other things if it's liquid. Is there mucus or blood present? What's the volume of the specimen? Do I need to centrifuge it before I plate it? Is the fluid clear or is it cloudy? Is there worms or segments of worms present in the stool? If it's an anaerobic culture, do you have a very foul odor or sulfur granules? And we'll talk about that a little later. But these things indicate that an anaerobic organism is present. And so if we do a macroscopic observation of the sample, we can determine if the sample is adequate, any special tests that we need to order, any special handling that we need to proceed with. And once we've done the macroscopic observation, we can do a microscopic observation. So here we can determine the quality of the sample. For urine samples, we could look at it under the microscope. For sputum samples, we could assess quality there. We can give a physician a presumptive diagnosis. Do we see gram-negative bacilli or do we see gram-positive cocci? So this can be helpful for treatment and when we're setting up media, when we're selecting media. So your primary inoculation, this is the types of media that we will inoculate that will give us the best possibility of isolating our pathogen. There are non-selective media which support the growth of non-fastidious organisms or not slow-growing organisms. An example is sheep blood auger. It doesn't select for the growth of any particular type of organism. It grows everything. Selective media. This is a type of media that will inhibit the growth of one type and allow another type to grow. Differential media allows us to differentiate between similar organisms. So sheep blood auger is not only is it non-selective, but it's also differential. It allows us to see different hemolysis patterns. Some types of organisms will lyse the red cells in this blood auger, and that helps us determine what type of organism we might be working with. So it allows us to differentiate between similar organisms. McConkey auger, we talked about this on the first day. We talked about lactose fermentation. McConkey's auger allows us to differentiate between gram-negative bacilli. Are they lactose fermenters or non-lactose fermenters? Enriched media, these have added nutrients to allow the growth of fastidious organisms. So fastidious organisms mean hard to grow. They need some extra nutrients. So chocolate auger is our most common enriched media. We can also use an enrichment broth. Pregnant women are screened for group B strep. And to do that, they swab the cervix and take the swab and place it in this limb broth. And the limb broth is incubated for 24 hours, and it will inhibit the growth of normal genital flora and allow group B strep to flourish. And that way we can isolate it better. Then we'll take the limb broth and put it on primary culture media plates and see if group B strep grows. And then there's also broth media that allows for small numbers of organisms or anaerobes or microaerophils. It's sort of a catch-all. Your thioglocolate broth is your catch-all broth. So here's an example of when you get a sample, what do you do with it? How do you process it? What kind of media do you put it on? Do I got to do a gram stain or not? Okay, so here's an example of, let's look at spinal fluid. So I got a spinal fluid from the floor. I've got to do a gram stain on it. I've got to set up a blood auger plate or sheep blood auger plate. I've got to set up a chocolate auger plate. I've got to set up a thioglocolate broth, and then some other notes. Cytocentrifuge recommended for gram stain. So I stick it in a cytocentrifuge. Anybody know what that is? What's it do? Right, so you put a couple of drops in this little bowl, and it centrifuges it, and it concentrates that sample into a little dot on your slide. And that's what you'll gram stain and look at. So it concentrates a sample for a gram stain. All right, so that's just an example. I'm not going to make you learn what plates to inoculate for different samples, but just realize that a variety of plates are inoculated, and it changes depending on the source because there's different pathogens at different sources. Okay? So specimen preparation, direct inoculation. So these are the different sample types that you will plate directly on your plate. Just like we did last week, we took a swab and put it at 12 o'clock on our plate. So pus, urine, sputum, bodily fluids, you would do the exact same thing. You put the sample right on the plate and streak it out using your quadrant streak to isolate. If it's a liquid and there's large volumes, then you want to centrifuge it. Swabs, if you have swabs, hopefully you'll have two. So one is used for the direct smear and one is used for the culture. And then tissue, if you've got tissue, you've got to grind it up using a grinder. And then you would, I take a swab and poke around at the ground up tissue and put it on my plates. So here's the different isolation techniques, and they should be familiar. We've got your urine streak, and then we've got your quadrant streak. So both of them allows us to quantitate the amount of organisms there, and we'll learn how to quantitate growth on plates today in lab. Okay, once you've plated your specimen on your media, it's time to incubate it. So most cultures are incubated between 35 and 37 degrees. That's body temperature. Depending on the type of sample, you may put it in a certain type of atmosphere. So you may have an aerobic incubator, you may have an anaerobic incubator, or maybe you put it in a bag with a capsule that makes the bag anaerobic. Capnophilic, you may have a CO2 incubator that pumps CO2 into the incubator, or you can use a candle jar where you put the plates in a jar and put a candle in there and light it and seal it up, and the candle will convert all the oxygen to CO2. And then a microaerophilic chamber will have a decreased amount of oxygen present in it. Time required, most cultures are held 2 to 3 days. Some of them are held for longer if the organism takes longer to grow. And so if you isolate an unusual or slow-growing, hard-to-grow organism, you may need to use some kind of specialized media or growth conditions for that. And we'll talk about those individual circumstances when we get into the organisms. Okay? So once we incubate it, 24 hours later, we come back, and we're ready to read our culture plates. Okay? Or culture workup. When you first look at the sample, you want to take note, what is the specimen source? So in my mind, I'm going to think, well, what is normally present at that site, and what is a common pathogen for that site? So if I pick up a urine culture, I'm going to expect different pathogens than I would a respiratory culture. Which media is demonstrating growth, and what's the purpose of that media? Is there only growth on my chocolate agar, and all my other plates have no growth? That indicates a fastidious organism. All right. If there's organisms growing on my McConkey plate, that indicates a gram-negative bacillus because it selects for the growth of only gram-negative bacilli. Does it require a full workup to genus and species? Would a just genus suffice, or do I need to send it out for additional testing or perform additional testing to find out what the species is? And that all just depends. So our routine specimens have standardized procedures. Wherever you work, there's going to be a procedure for how to work up a sputum sample and what you have to do depending on what growth you see on your plate. So those are all standardized. You'll have something to follow. We're not going to talk about non-routine specimens. Your book talks about how to handle something that's not routine. We're not going to talk about that here. Okay, so once you've looked at your culture, now you need to communicate your findings to the physician. You want to report preliminary results when they're available so the physician can go ahead and treat the patient as soon as possible or change treatment if necessary. Clearly interpret results to avoid confusion. You want to avoid technical jargon that microbiologists understand, but physicians may not understand. Avoid abbreviations like GPC. They may not understand GPC. What is that? Critical values. If you have a critical value, for example, if you find yeast in a spinal fluid gram stain, that is a critical value, and it needs to be reported to a nurse or a physician immediately. And then you have to document who you called and what time. So each facility has a critical value list. So depending on what those are, you would call if it's a critical value. So here's just some examples of critical values. If you see organisms in your blood culture, in your spinal fluid, those kind of things. All right, you need to take a break. I see some sleepy eyes. Let's take a quick break. All right, microscopic exam of infectious material, infected sites. All right, take a look at your objectives. There's not too many. Oh, that's good. A 75-year-old man with a history of chronic obstructive pulmonary disease, or COPD. He's a heavy smoker and some alcohol abuse. Comes to his physician with fevers, chills, and a productive cough, so he's coughing stuff up. Sputum samples are collected and sent to the lab for a direct smear and culture. Blood cultures are also drawn 3 times within 24 hours of admission. So the direct gram stain, direct smear, is on plate 14. So let's find that in our book. It should be near the end of the chapter. There's some images. Page 143, plate 14. What do we see here? What are those big pink blobs? White cells. Do you see any squamous epithelial cells in that field? Do you think it's a good sample? All right. What kind of organisms do you see? Gram-positive bacilli. What do you notice about their arrangement? They're in pairs. They're diplococci. Okay. Do you see anything unusual also kind of around them with this? A halo. What does that indicate? A capsule. Interesting. All right. Notice that they're almost kind of long bacilli, or long cocci. Almost like ants with no legs. Yeah. They're kind of stretchy cocci. Interesting. Okay. All right, so the sputum culture produced heavy growth of an alpha hemolytic colony, and the blood cultures yielded similar results after 24 hours of incubation. So what do we notice? The same organism is growing in the blood culture and in the sputum, isn't it? All right, so whatever's causing his cough is now getting into his bloodstream, making him septic. So that's not a good thing. It's a gram-positive cocci in pairs with a capsule. They're stretchy, ants with no legs. That makes me think of also of the growth on the plate. It's alpha hemolytic colonies, which means it destroys the red cells, not completely but partially. We see a greening around the colony, and I have an example in lab so you'll be able to see this in person, and that is characteristic of Streptococcus pneumoniae. So based on that gram stain that you would give, and the physician will have those gram stain results long before he has the results of the culture. Okay, the physician can go ahead and start treating the man for a gram-positive cocci. Where's my clicker? Where did I set my clicker down? Here it is. Okay, so the gram stain gives us a way to rapidly give the physician a result so he can act on it, or he or she can act on it. They can treat the patient with appropriate antibiotics based on the morphology of the organisms that we see. All right. 88% of the time, the physician has the correct presumptive identification before they get our gram stain results. Or maybe that is with our gram stain results. Based on our gram stain results, they have a presumptive idea of what the causative agent is. And 88% of the time, they're correct. All right, so they can initiate the correct kind of treatment. But those that other, was it 12% of the time, they may their presumptive identification is incorrect. So that's where the culture comes into play, and we can give them the correct identification, and they can change the treatment if that presumptive ID is incorrect. All right. So when we get the sample, we've got to prepare our smear. All right, so smears from swabs, you just want to roll the sample over the slide so you get all contact with the whole swab over the slide. Because if you have to prepare a gram stain, you always want to get 2 swabs. If not, then you want to do the gram stain last. And here this has you sweep the swab over the entire surface of the slide. This is like the frosted edge where you would label. I don't like to do that unless my sample is very thick and viscous. If you smear it over the entire slide, you're going to be there all day looking through that gram stain. So you want to keep things small, a dime size ideally, in our target circle, so you're not searching the slide all day long. Okay. Preparing infected materials for visual exams. So we'll talk about like when we use stools to look for parasites, when we prepare wet prep examinations. You might mix the swab with some saline. Hopefully it comes that way from wherever it came from because the nurse collected it properly. You might need to cytocentrifuge the sample if it's a fluid. You may need to prepare a smear if it's some kind of a fluid like pus or peritoneal fluid or something like that. You just take a drop with a pipette. The one thing about drops and pellets, you don't want to make them very thick because they can rinse off very easily during your gram staining. So you want to be sure to make them thin preps. Tissue, you would do a touch prep, a gentle touch of the tissue on the top of the slide. So smears from thick liquids or semi-solids. You want to immerse the swab in the specimen for several seconds and then prepare a thin spread on the slide. If you have granules in there, you want to crush the granules maybe between 2 slides. Here's an example of the 2-slide technique where you take a slide and place it on top of the drop. Gently press it down. I don't press it down. You can kind of feel it like gloop to each other. Gloop. Yeah. It kind of, I don't know. Yeah, you can feel it. Did you do this in hematology? Did you do a 2-slide technique? Well, you can feel it like grip onto each other. So you don't want to press it together because that could damage the cells. So you'll feel it. It's kind of like a suctioning thing that happens here, and you just pull them apart gently, and it makes a pretty good smear. Smears from thin fluids like spinal fluid. You want to make your target circle because especially if it's clear, it's not cloudy, it's very thin. Once you make your gram stain, you don't know where you put it on your slide, and you don't know where to focus. And it's hard. We had that experience, didn't we, the first day? So it's important to have that target circle. And then cytocentrifuge preparations. This concentrates the fluid into a circle, and it allows us to the protein can be filtered out through a pad so that we can see the background a little bit easier. I tried to find a picture of a good cytocentrifuge, but I wasn't able to find one. So basically it's like a little cup. A little cup kind of like this, and it's got a tube connecting from it, and then your little filter is here. So you put your drops of your sample in here, and when you centrifuge it, it pushes it out this way through the filter, and you have a slide right there. So it concentrates it right here on the slide. That way the probability of seeing an organism is much better. Let's see. You can fix it with alcohol, or you can air dry it and fix it. This is something else. If you do a pellet or a cytocentrifuge and it's pretty thick, you can take a loop and kind of smear it out, and that way it won't be too thick when you gram stain it. You'll have a thick area to look at, and you'll have a thin area to look at. So we talked about several different types of stains. We did the simple stain, which just allows us to see basic shape and forms. We can't differentiate between organisms. Everything was blue. Everything is stained the same color. Differential stains allow us to differentiate between organisms. So the gram stain, the coloring of the different components are based on the makeup of the cell wall, right? So which one stains purple? Gram positive. And why do they stain purple? No, they don't. Peptidoglycan. They have a thick layer of peptidoglycan. And what about pink organisms? Very thin smear. They have a thin layer of peptidoglycan, and they have an outer cell wall, which allows it to decolorize very easily. There's probe-mediated stains, so we can attach a color to an antibody, and if the antigen is present in the sample, the antibody will bind. And if we stick it, say it's a fluorescent probe on that antibody, we stick it under a fluorescent microscope, and the antigens will shine. Or we could use a DNA probe, so a probe that's directed against a specific sequence of DNA in a sample will bind. And we can use different stains to detect that DNA, stains that bind DNA. We talked about acridine orange will bind DNA. So we can detect very, very small amounts in that way. And we won't really talk about those too much. So gram stain principles. Is everybody okay with this? Gram stain principle. Why? The principle is why it occurs the way it does, or how a positive is positive. Okay, and that's important. We're going to divvy out some immunoassays, poster topics for you. And I have a lot of students kind of get confused between the purpose and the principle. So the purpose tells me what. It answers the question, what. And the principle answers why or how. So how a gram-positive organism stains gram-positive, or how a gram-negative. You got to tell me. Basically, a thick layer of peptidoglycan retains the primary stain, crystal violet, and complexes, the stain complexes within that layer, and it resists decolorization. And that's the principle of the gram stain. Gram-negative organisms are unable to withstand decolorization, and the crystal violet iodine complex is washed out. So that's how the gram stain works. The purpose is it allows us to differentiate between organisms based on their cellular composition. Okay, I'm going to skip over this because we did the gram stain last week. It's pretty simple. We talked about some limitations and some sources of error. If you make your smear too thick, or you leave your saffron on too long, or you over-decolorize, or you under-decolorize. What happens when you over-decolorize a gram stain? So you leave the alcohol on way too long. What's going to happen? Gram-positive things will appear gram-negative. What if you under-decolorize? What if you don't decolorize enough? What's going to happen? Very good. And you might have some precipitate. That complex can form on all that proteinaceous debris. And if you don't rinse all that out, you're going to have a lot of debris left, all those artifacts left over. All right. These are the different types of microscopes. We use the compound microscope. Okay, terminology for direct exam. It's important to standardize how we talk about microbiology, how we communicate results. And that way we can be on the same page not only with our coworkers but also physicians and nurses. We can communicate with people in different countries. We can use the same computer systems. They're all set up basically the same. The microorganism description, or how we describe an organism, can give someone a presumptive identification. So that we suspect a specific type of agent. So when I say gram-positive diplococci, lancet-shaped, which is what our case study showed, long, stretchy gram-positive diplococci that look like ants with no legs, that makes every microbiologist in the world think of the same organism, Streptococcus pneumoniae. So it's important that our terminology is on the same page. So here's just an example from your book of certain terminologies. When a microbiologist hears gram-positive cocci, they start thinking of these common organisms. All right, Staphylococcus, Streptococcus. All right, when I hear gram-positive cocci in pairs, I think more Streptococcus. Here is gram-positive diplococci, lancet-shaped. Notice Streptococcus pneumoniae is what you think of. Gram-negative diplococcus. I see Neisseria and Moraxella cataralis. Gram-positive cocci in chains, I think of Streptococcus. Gram-positive cocci in clusters, I think Staphylococcus. Let's see, what does it have here? It says microaerophilic Streptococcus, viridin strep, and Staphylococcus. I want you guys to think of staph. Clusters, staph, chains, strep. Okay? All right. Okay, so direct exam terminology. And this is, you can comment on all kinds of things when you're doing a direct exam. I just want you to be concerned with white cells and epithelial cells. If it's a sputum, we comment on epithelial cells. Mononuclear cells. Typically, bacterial infections, you see segmented neutrophils. So if all your white cells are mononuclear, that could indicate a chronic infection. So you would want to notate that. If all your cells are mononuclear, like all lymphs, all monos, you want to make a note of that, because commonly they're just neutrophils, PMNs. You might comment red blood cells. I commonly don't unless it's like a spinal fluid, because that indicates a traumatic tap or a hemorrhage. But otherwise, I don't really note red blood cells in my smears. I may note it on stool, though, if I just look at the stool and I see blood. Okay, so examination of prepared material. We kind of got a hint of this last week. We quantitated organisms. All right. Let's see. Commonly, when we have an infection, we have greater than 10 to the 5th colony-forming units per milliliter. And that sounds really complicated, but when you look on the gram stain, you see 1 gram-positive cocci. 1 individual gram-positive cocci. That is a colony-forming unit right there. Okay? This one single organism will divide into 2, and divide into 4, divide into 8, and 64, right? So eventually this one organism, this one microscopic organism, will turn into that colony that we see on our plate. So an infection commonly has organisms numbering 10 to the 5th. How many organisms is that? How many zeros? 1, 2, 3, 4, 5. That's 1,000,000 organisms. Okay, so it's very easy to see organisms when there's an infection present. There's a lot of them. Okay, there's 2 types of infections. We can have monomicrobial. What do you think that means? one organism is causing the infection, or you could have polymicrobial. And what do you think that means? Many organisms are causing the infection. So there's certain types of sites that commonly, perirectal abscesses and anaerobic abscesses, or those kind of things. They'll commonly have many types of pathogens present. Those are a little more difficult. Okay, so when you first put the slide on your microscope, you kind of want to look around at your background material. Is it homogeneous or heterogeneous? You want to do this on 10x. Do you have, is a lot of mucus centralized in one area, or is it a nice slide where everything's distributed properly? Sometimes with sputums, you'll have big, thick strands of mucus where all the white cells are gathered, and then you'll have big empty spaces where there's nothing really. So you want to make sure to look over the whole slide. You're not going to be able to make a perfect smear all the time. So in that case, you want to make sure to scan the whole slide, and you're not just looking at one spot, because pathogens may not be distributed uniformly. Is there evidence of normal flora? Do you see epithelial cells? Do you see a lot of bacteria but no white blood cells? That indicates a poor sample, right? If there's no white cells, then you don't want to think infection. If you see a lot of organism and no white cells, that doesn't quite correlate, right? If you've got an infection, you should have some white cells. Okay, so once you do your 10x scan, you might scan on 40 or 60x and quantify your squamous epithelial cells. This is done on sputum samples only, okay? Now, you might have a facility that changes that. You might look at squamous epithelials on other types of specimens, but for our student lab, I want you to quantify epithelial cells on 40x on sputum samples only, and that's in the procedure. Once you've done that, you want to go to 100x and quantify white blood cells and each morphology of organism that you see. Okay? You want to ask yourself, is this a sterile site? Is there more than one organism present? Does that indicate contamination? Okay, you might have to reject a sample if you see more than 3 different morphologies. Don't overinterpret the findings. Don't write a book on each different morphology that you see. Start grouping them together. If you see a lot of gram-positive cocci, just say gram-positive cocci. Don't tell me clusters, chains, diplococci, and tetrads. That's a lot. Your hand's going to get tired. Okay, group them together. If you're unsure, wait for additional testing or staining. You guys don't have that liberty. You'll have to give me the answer right now. But in the real world, you can go ask someone, what do you think about this gram stain? If you're unsure if it's trash, if you just see very rare gram-positive cocci, don't call it. Wait until the next day. Maybe put a question mark next to it. Don't put it in the computer and say, hey, you know, I thought I saw this, but I'm not sure. Evaluation of antibiotics. We're not going to talk about that. Special handling. So you might need to modify some tests if you see a certain type of organism. So Haemophilus, if you suspect Haemophilus, you would work with the chocolate agar, because Haemophilus only grows on chocolate agar. If you are suspecting a slow grower, you might have to increase the incubation time from 3 days to 7 days. You might need to add special media if you think that there's a Legionella present or a Mycobacterium. If you think there's something else there that requires a special kind of media, you want to add that. You might need to change a temperature or atmosphere. We're not going to go into a whole depth of adjusting, but just be aware. So grading and classifying materials. So this allows you to prevent culturing normal flora or contamination. So if you get a sputum sample, commonly you'll do a gram stain first, and depending on whether or not the sample quality looks acceptable, then you'd go ahead and set up the sputum sample. If not, then you would reject it and ask for a recollect. So it allows us to assess our sample's integrity. And how to quantify these things are in our procedure manual. When we're evaluating sputums, this is the quantification scheme that we're going to use to determine whether or not we're going to accept or reject a sputum sample. There's different ways of assessing sputum quality. So when you go to a clinical facility, they may have a different way of doing this. So with us, if you see less than 25 squamous epis and more than 25 white blood cells, we're going to call that an acceptable sample. Greater than 25 squamous epis, not a great sample. There's a lot of skin contamination there, but we have a lot of white cells present, right? So we're going to accept that. That's mixed secretions, but it's an acceptable sample. Here we've got greater than 25 squamous epis, so it's contaminated, and we've got less than 25 white blood cells, so there's not a lot of white cells there, so we're going to reject that one and ask for a new culture. All right. Lastly, once you have growth on your plate, you want to correlate that with your gram stain results. So, do they match? If you have a gram 4+ gram-positive cocci in your direct smear, you want to make sure that in the culture you have 4+ gram-positive cocci organism growing. All right? Make sense? So if you've got a gram stain that has a gram-negative bacilli, and when you look at the plates the next day, a gram-positive bacilli is growing, what do you think happened? The gram stain shows a gram-negative bacilli, and the growth on the plates shows a gram-positive bacilli. Ooh, decolorized your stain too much, right? What if you've got a gram-positive cocci on your gram stain and a gram-negative rod, but you've only got a gram-negative rod growing in your culture? So we've got 2 morphologies on our gram stain and only 1 morphology on our culture. So we got something's not growing, right? The gram-positive cocci. Maybe you need additional media. Maybe you're not growing. Maybe it's an anaerobe and it's not growing on your aerobic plates. Maybe it was precipitate and you confused it with an organism. Maybe the patient's on antibiotics and those gram-positive cocci are dead and they're not going to grow. So there's some things that might happen. So you always want to take a look at your gram stain and make sure that matches with the growth on your plates, because that'll tell you, I need to do some more digging. You know, I need to find out why. So here's an example of a report. So it's a respiratory culture. It gives us a source. It's a expectorated sputum. The microscopic exam, we saw contaminating bacteria, yeast, and a lot of epithelial cells. So we saw a lot of contamination, right? Not a great sample. We also saw gram-positive diplococci, consistent with pneumococci. I don't say that, but gram-positive diplococci suggests the Streptococcus pneumoniae. And here it says that they called to the doctor. You may or may not do that. And then under here we have the results of the respiratory culture. So this is what grew on the plate. All right? Sinus, ethmoid contents. The microscopic exam shows a local, we're not going to comment on purulence or light local materials or red cells, but they saw gram-negative bacilli. Okay. quality control in direct microscopic exams. So we did a little bit of this before we gram stained last week. We had a prepared slide that had a known organisms on it. We knew that it should stain gram-negative bacilli and gram-positive cocci. So if we keep up with our QC, and there's a whole range of different quality control methods in microbiology, we can make sure that our smears, our gram stains are working the way they should, that our culture interpretation is working the way that it should, that our biochemicals are working. It could help us catch any discrepancies if something's not quite adding up the way it should. It allows to provide education and improvement. It helps us to track technologists who make errors and allow us to correct those problems if we need to. Okay. Any questions there? Those are those two chapters. Take a look at the color plates at the end of the chapter. See if you can find the organisms. Just look at them first and try to find the organisms and characterize them. And then check to see if you were right with the caption underneath. Okay. You can do that on your own time. I'm not going to make you do it here. I want to draw for these little immunology posters that we're going to do. So what you need to do is get with a partner. Find a partner that you want to work with. I think somebody's going to be left out because we're missing Kara. Does anybody want to work with Kara? Who's left out? Is there someone left out? Somebody. All right. Kara's partner. All right. We can draw randomly these topics. I'll start on that side next time. Y'all are partners, right? Write you and your partner's name on the topic, and we're going to return it to me. I'll post it on Blackboard when we put it together. Who's going to choose here? You got two? No. All right. All right. Now, put your names on there. What is that? Under content, I have the immunoassay posters. I have the rubric, so you can see how I'm going to be grading each of these posters. I have the immunoassay table, which you'll fill out after the posters are posted. And this will give you a nice little table that summarizes all these different immunoassays. So when you're studying for your Board of Registry, you can hopefully go to this table and it'll sum things up. You don't have to go digging around in a textbook. And then expectations of the presentation. So this is what I'm looking for. So use these expectations and use the rubric as you're building your poster with your partner to make sure that you cover everything. Come on now. So there's the table. You can type it up in Word if you want to once you collect the information from the posters in the class, and type it up all nice and neat, or handwrite it, whatever. Okay. This looks like the old. This is my old expectations. I posted the wrong one. I'll have to repost it. I have a hard copy here. So last year we did presentations. I wanted to avoid that this year. So let's see. Posters are due on Monday, February 9th. So that day, I'm not here. Okay, it's a integrity lecture on that day. So you'll be here for chemistry anyway, so just post them up there on the bulletin board. Let's see. The immunoassay table is due Monday, February 23rd, so that's the date of the exam. So poster specifics, use an 8.5 by 11 inch poster, and that's just a page this size. Okay. You just use one of these blank pages if you want to. Just simple. I don't want you to spend a lot of money. Use a 12 or 14 inch, 12 or 14 font. Use Times or Calibri font so that we can read it. Nothing real fancy and elaborate. Okay. Use clear and understandable graphics. So I want to see what's going on in that reaction. So most everybody in here, I think, is going to have an antigen or an antibody or a conjugate or a substrate or something, just depending on what method you've got. So draw a picture or build a picture with a computer of the reaction that's happening. So some of you might have a cassette, a test that happens in a cassette. I don't want a picture of a cassette with 2 lines and another picture with 1 line showing a positive and negative. I want a picture like in your textbook on in Chapter 10. There are examples of the different immunoassays. So say you've got a solid substrate for your assay. Say you want to detect an antibody. I definitely want a key somewhere, maybe in the very bottom. You know, it doesn't need to be big. Say the patient's antibody is what you're trying to detect. Maybe you're detecting hepatitis B antibody. Okay, well, how are we going to capture that antibody? Maybe we have an antigen on the bottom of our well. This antigen is the hepatitis B antigen. It binds directly to our antibody, doesn't it? So we add the patient's plasma. If the antibody is present, it binds to our antigen. Now we got to see it somehow, right? So we got to put something that's labeled in there, maybe a fluorescent tag, maybe it's a colorimetric method. So we got to add something. So maybe you add a conjugate. Here's an antibody conjugate. It's an immunoglobulin that binds immunoglobulins, an anti-human immunoglobulin. Makes sense? Yeah. So it's a conjugate that binds antibodies, and it's labeled with some kind of enzyme. Okay? So we add that, and if the antibody's present, it's going to bind. Okay? What happens next? We got to add something to activate the enzyme, right? What do we call that? Substrate. A substrate is something that an enzyme works on. So this is what actually produces or causes the color. The enzyme produces the color, but the substrate is what it acts on. Okay? So if if our patient's antibody is present and our conjugate is present, our substrate binds and fluorescence is emitted, or a color is emitted, or a band is formed, or something happens. Okay? And if it's negative, if the patient doesn't have the antibody, then no antibody binds, right? We add our conjugate, and what's it bind to? Nothing, because there's no immunoglobulin to bind to. So our conjugate's floating around. Do we add our substrate? There's an important step right here. It's your wash step, right? You've got to wash between adding your conjugate and adding your substrate. Otherwise, what happens? Your substrate will bind here, and you'll get a false positive, right? So you got to wash away anything that's unbound. Okay? So in a negative, we'll wash away our conjugate and add our substrate, and nothing happens because there's nothing to bind to. So this is a very important wash step. Okay? And the solid phase, all that is, the antigen bound to the solid phase, that just keeps it in the cup when we wash it. It's bound to the bottom of the micro well, or it's bound to the little strip, or whatever, and it keeps things where they need to be. All right? Any questions? It's immunology in a nutshell, basically. Okay, so if you've got any questions about your method, if you're like, I don't understand this, definitely email me. Check out your immunology textbooks. I know you kind of had a Pearson textbook, and those aren't the best. They're kind of directed at, I don't know who, but they put things in very difficult terminology. There's some good textbooks up here. Turgeon is the one, I used him before. It's really good. It kind of breaks things down a little bit easier. What else? I thought I had another one. I might have one in my office I'll bring down. Yeah. So use one other source. Here's the Stevens. This was mine that I used in school, so I prefer it, but it's good too. So use one other source besides your micro book. Immunology book is a good source. Okay? What else? Keep it simple and keep it direct. What's necessary? What's absolutely necessary to put on your poster? Okay? Your poster has to include the type of the assay. Okay? So that's going to be your title. Just the type of assay that I assigned you. Okay? The principle of the methodology. How does the method work? How does a positive become positive? How does a negative become negative? So you're going to describe to me what's going on. I want you to put it in your own words so that you understand it. Okay, these guys have had a lot of school. They know what they're talking about. I don't care what they want. I don't care how they say it. Put it in words that you and your peers understand. Okay, talk to each other. You know, collaborate with each other. I don't care if you get together and study together. I would like you to study together. Find out how Mallory thinks of a reverse passive immunoassay. You know, she might explain it in a way that you've never heard it before. That just clicks. So use each other. Bounce ideas off. Just not on tests. Okay. And then include an invalid result if it applies. Some of you guys, it doesn't really apply, but I think I gave somebody a Western blot. That's the one I was thinking of. In those, we have an indeterminate result, and so that's important that we know what's an indeterminate result compared to a positive and a negative. So that's the one I was thinking of. And then some specific examples of the methodology used for infectious disease. Is that test used for RPR testing for syphilis, or HIV testing, or mononucleosis, or strep pyogenes, which causes strep throat? What's it used for in microbiology? Okay, and just a couple examples. Two is fine. If you can't find any more. Any questions? On the back of your poster, put a bibliography. Okay, I'm not fancy. And then one last thing is multiple choice questions. I want 4 multiple choice questions with 4 answers. Write it. Don't write it like someone is standing in front of your poster reading your poster. Write the questions so that I can put it on the exam. So they don't need to look at your poster to know it. They, having read your poster, will be able to answer the question. Okay? So don't say, what is the pink square on the poster? I can't use that. But what is the most common use for reverse passive agglutination or whatever. An example of it in microbiology, whatever. So 4 multiple choice questions, and I'll put them on the exams. So if you want to share with each other your questions, you can do that. And each of you will submit the questions individually in Blackboard so I can give each person a grade. Okay? Make sure you spell check them. Make sure it's a prepared question, that it sounds correct, sounds like it makes sense. Bounce it off other people if you have to. Okay, that's it. Not bad, right? Not too bad. And those are the only questions from this chapter that will be on the exam. I'll use y'all's questions. But I won't give credit. It's 4 points for the multiple choice questions, and I won't give credit if it's poorly worded or you don't have enough answers or something like that. So as long as it's a good question, it's easy points. Okay? Question. The rubric is on there. I've got content or principle is worth 12 points. It's divided in content, organization of material, mechanics, and audience awareness. So 3 points is the best you can get at each category, and 2 points, 1 point, and 0 points if you didn't complete it. So I give an explanation of how you can get 3 points there. So make sure you follow that. All right. Visual aids, your pictures, that's worth 3 points. And then followed directions, your bibliography, and your multiple choice questions. So it should be a pretty easy grade here. Okay. When are we done here? 2:30?